All right, everyone. Welcome. So here we are. We're gonna get started. You had a chance to put your location in. You can see where everyone is from back on our shared Zoom screen here. And we'll get going. So I've met some of you before long ago. I'm Sarah Saladay, formerly Sarah Silverberg, and I've partnered with the University of New Hampshire Leitzel Center to be here with you today. I'm coming to you from Littleton, New Hampshire, where I live. I'm hanging out in my basement workroom while my children are sleeping, and I'm super excited to be here. Jen Bourgeot is also with us today. Jen, do you want to introduce yourself?
Sure. My name is Jennifer Bourgeot, and I am coming to you from the University of New Hampshire Leitzel Center as well in New Hampshire, United States of America. And it is a beautiful rainy day here.
So just a couple of reminders before we get started. This is not an official protocol training, but we will be highlighting several parts of the carbon cycle field protocols modules today. And you can check out the full e-training modules on the Globe website after the workshop. There'll be lots of opportunities to participate in today's workshop, kind of virtually. So first, if you have any questions, feel free to type them into the chat. Jen will be there to answer them. She's also going to tell you another place where you can answer your questions, get your questions answered. And there's also many linked activities. So just like the Padlet you just participated in, you'll see a link pop up in the chat, and you can open that in your own browser window and participate in the activity. And then when you're done, you can return to the Zoom window and we'll have more information about what you've all just done, and we'll kind of share the screen so everyone can see everyone else's work. And of course, please don't hesitate to let us know if we need to slow down a little bit. Sometimes I forget and speak too quickly. But also keep in mind the workshop is being recorded so that you can refer back to it later if you feel like you've missed something. So you'll see another link coming up in the chat now. It's for something called Mentimeter, and it's a place for you to type your questions so that Jen and the rest of the Carbon Cycle team will be able to answer them throughout the workshop. And if we have time, we'll go over them at the end. And if we don't, you should check in on the protocol Q&A session on Thursday to get your questions answered. And hopefully you did have a chance to participate in the Carbon Cycle introductory module, e-training module. And if you have any questions about that, you can also enter those into the Mentimeter link. But if you didn't, that'll be available for you on the Globe website. So let's just start at the beginning. Why collect carbon cycle data? We all know that scientists need to collect carbon cycle data in order to understand climate change. And in particular, our focus is the carbon cycle in terrestrial ecosystems. So when students and teachers collect carbon cycle data with Globe, they kind of contribute to this better understanding of the relationship between carbon storage in plants and surface climate. As all good teachers do, we're going to start our workshop today with a research question and kind of focus our energies on answering the research question by using the Globe materials and experimenting with some of the different activities and protocols that sometimes cause confusion. So our focus question is, how do differences in tree species and sample site affect carbon storage near where I live. So how do differences in tree species and sample site type affect carbon storage near where I live? So what do we need to know, and what do we already know about this question? So we have a link for you today in something called a Jamboard, where you can make direct edits. And right now we're going to go over there and write sticky notes in columns on what we know and what we want to know. And then we'll come back to it at the end to type a little bit about what we've learned. So your link's going to pop up in the chat. You drag that to your browser window. And then you'll see the Jamboard pop up, and you want screen 1 or frame 1. And that's, you can adjust those at the top of the page here. And you can just start putting in sticky notes about what you know that'll help you answer our question, and then what you want to know that'll help you answer that question. So everyone can take just a few moments to do that now, and when you're done, you can come back to the Zoom screen and we'll take a look at what everybody has entered. Well, good. I'm seeing people enter some ideas, and then you can drag those sticky notes wherever you want to put them. If you've forgotten the question, you'll see it's right at the top of the page. Lots of great ideas coming in. I see that people understand that carbon is stored in living things, that larger, older trees hold more carbon, that the more biomass in a tree, the more carbon is stored, that a forested site will hold more carbon than a site that contains buildings. It's like there's also some really important things that we're going to need to find out, some of which are going to help us directly answer our question, and some of them are a little bit less important for answering this specific question, but may be also interesting to study when talking about the carbon cycle. So how do you even identify different species and which species are in your area? How invasive species might impact the carbon storage of your area? Do different tree types store different amounts of carbon? Ooh, a very in-depth question about factors like mycelium and soil nutrients and their impact on carbon in trees. Do conifers store more carbon than hardwoods? Does the site specifically affect the growth? The impact of local climate events? And the relationship between age of the tree and how much carbon is stored. So this is a really good platform to get started with our question. And this is how I always try to approach it with teachers and students when we do trainings and then also in the classroom, because it activates prior knowledge and it gets us to this point of like, why are we even going to do some of the activities we're going to do? So people can certainly keep working here. If they've got a few more ideas, you can keep adding them. We're going to come back to this slide towards the end. So today we're going to focus a bit on kind of narrowing things that are difficult to understand, but still covered in the carbon cycle materials that we've created. So up here on our field protocols flow chart is the carbon cycle introductory activities. So if you need more background on carbon cycling, this is a great place to go. Some teachers and trainers already have lots of experience, as do some students, depending on what grade level you're teaching or what age level. After you have a background in the carbon cycle, there's some field learning activities, and we'll talk about some of those today. And then you would be ready to move on to protocols, so site selection and then site setup. And then depending on the kind of environment you have, you might do tree protocols, shrub and sapling, and or herbaceous protocols. And then once you've collected all your data, you'll move on to data entry, data analysis, and assessment. So today we're going to focus on site selection, allometry and biomass unit activities, and data analysis. So just the order we're going to go in today: protocol site setup, comparing standard and non-standard field sites. We're going to participate in part 2 of biomass units and part 2 of the allometry activity. And then take a look at some data analysis in a spreadsheet, kind of digging in a little bit. So how will you calculate carbon? First, you have to measure tree circumference. Then you need to convert it to diameter at breast height, because that's what the way that scientists have used, have been able to calculate biomass. Biomass is related to carbon storage in an individual tree, and then it can be scaled up for an area. What you see coming in now are how we go from one step to the next, and that's going to be the focus of our work today. So we're going to actually start kind of towards the end and take a look at sample sites and do a little bit of thinking about what sample sites we have, well, you have at your location, so that you feel confident in which protocols you're going to choose. Site selection is important. Of course, most of you have been involved in GLOBE for some time, and so you're familiar with different sample site setups, depending on if you're doing precipitation or land cover. So carbon cycle can be very similar to the land cover sample site setup. You can do a 30 by 30 meter square, but you don't have to. The most important part is that you have an accessible area That's at least 15 by 15 meters or 225 square meters of contiguous vegetation. So that might be forest, shrubland, grassland, or it could be non-native. So it could be a tree plantation, and that's fine. And then you would also be able to do a kind of a non-standard site near your school. And that also needs to be accessible and have that same minimum size, but it could have some human interference, such as buildings, roads, etc. So we're gonna take a moment here. Jen's gonna put the Flippity link into the chat, and you can click over there and do a quick little activity on standard versus non-standard. So you're going to move the pieces around to make sure you understand what the different sites contain. And then we're going to think a little bit about your own sites and talk about our example site for today. So put the link in your browser and head over to Flippity. And you can do this work on your own and not look at my screen. Or if you're not wanting to do that, you can follow along with me. So I'm just organizing the components. If you've finished and want to see how yours stacks up, the one I'm working on, you can head back to the shared Zoom screen. If you have any questions, feel free to type them into the chat and we'll do our best to answer them. Jen's put a nice teaching tip in the chat. So feel free to come back to your own work if you're not quite done. But if you want to take a look now at our main screen, you see we simply divided two halves into standard and non-standard, and then move the pieces around until we had parity. You'll see that similarly, they both have the same minimum size and that they need to be accessible. But you'll notice the pictures look quite different from one another. On the standard site, we have that contiguous vegetation, often native vegetation, but it doesn't have to be. And on the non-standard site, it's vegetation interrupted by human-made structures. So now it's time for you to do a little bit of thinking about your own site. So is your site that you're going to train or teach on standard or non-standard? You're going to jump into a Padlet. You're going to type up a description of your site and put it under the standard or non-standard or I don't know column. And then give and get feedback to ensure you know which protocol e-training that you'll want to follow up with after this workshop. So there's an example of our University of New Hampshire College Wood site in there already. So if you're done with yours and you're looking at other people's, if you see any ways that you can help them, you can type in your comments. Okay, so it looks like some people have standard sites that are very, very standard, I would say. A national park plot, a forest adjoining a university, someone's backyard, that's a complete woodlot. The Notre Dame school playground is more likely a non-standard site. It's possible it could be standard, but it would have to be at least 15 by 15 meters or some other configuration of 225 square meters total, without any structures or buildings that would be taking away from the vegetated area. And that also goes for the question over here, the park with trees and grass and one sidewalk running through the middle. It kind of depends on the sidewalk. Does it seem like sort of a natural environment with one small path, or is it a very manicured environment? If it's a largely manicured environment, more likely you'll want to do the non-standard protocol. Standard is much easier to do with natural vegetation that's sort of been uninterrupted or unimpeded by human use. Like I said, with the exception of tree farms, which are planted to be contiguous vegetation. If anyone has more questions about that, they can continue typing those in the chat, and also maybe some of the, maybe Jen, or we actually have 2 other co-trainers on this call that are lurking in the shadows. If you see anyone's questions on this Padlet and you have the opportunity to answer them, that would also be helpful. So the site that I'm going to focus on kind of for the rest of the workshop, just as an example, is the training site at the University of New Hampshire. And this is kind of what it looks like. So we know a little bit about it because there's a sign as you enter. It's a natural area, 64 acres, so several hectares. And it was set aside land for research and enjoyment by students. And here's a side view, and this is kind of one trail that goes through. So this might be a sidewalk question. And then this dotted line is the green, this dotted green line is also the trail, but we actually put our plot over here so that it wasn't impacted by any of the trails. So that's something you could do too if the area is large enough. So where do we go from here? I know what my site looks like. I know that I'm going to perform a standard site setup protocol. And now I have to choose, do you have 15? Well, which protocols am I going to choose? What vegetation am I going to collect? So this is a decision tree that's available for both the standard sites and the non-standard sites. And the way it works is you read the question. If the answer is yes, you follow that arrow. If the answer is no, you follow the other arrow. So for our Collegewood site today, do you have trees greater than 15 centimeters circumference? We say yes. Do you have shrubs and saplings on greater than 25% of your sample site? And if you are looking carefully, you would know that the answer is no. And do you have herbaceous vegetation on greater than 50% of your sample site? And for our Collegewood sample, that is also no. It is large overstory trees with very few shrubs or undergrowth. So we're just going to collect data on trees for this Collegewood site and follow that through for the rest of the workshop. So here we are again. Most of us as adults know the relationship between tree circumference and diameter. But the How to Measure Trees activity on the Globe website for students gets at that concept and introduces them to this equation if they haven't had that experience before. It also teaches them actually how to use the measuring tape to pull it around the tree and at what height to do it, and sort of the precision and accuracy required to make good measurements. So it's a great introductory protocol for especially younger students or students who haven't had a lot of science experience. But right now, we're going to kind of skip ahead to biomass, because we know tree circumference, we can get diameter, but then we go to biomass. And what exactly is biomass? So the study of biomass. Biomass is the total mass of all living things within a given area. So all living things within a given area. That could be the biomass of a classroom or a field site or the globe. Biomass is 50% carbon by dry mass. So what I mean by that is if I took an area of grassland and I cut down all the grass in that area, I would be holding the biomass in my hand. And then I'd take it and I'd dry it out. And then I would mass it, and that would be its actual dry mass. And then 50% of that mass is how much carbon is in that grassy area, that herbaceous area. And it's important to kind of understand that as the link between biomass and carbon. It's also important to recognize that on a global level, vegetative biomass is a really important number in the carbon cycle. You can see the global carbon cycle diagram to the right here. Plants contain 560 petagrams of carbon, and that's a very significant number, although if you then compare that to soils, it's only just about a third. But you may have also noticed on the global carbon cycle diagram that there's no carbon cycle number for animals, and it's because the mass in animals just doesn't compare to the mass in vegetation across the terrestrial ecosystems. And what's really great about introducing these carbon cycle protocols to students on a local level is that local plant communities actually really provide important insights about carbon storage on that micro scale. That 560 petagram number actually comes from lots and lots of people studying terrestrial ecosystems and measuring the carbon around the world. So in order to get at this concept, we use the biomass units activity. Part 1 is really conceptual for students. It has them calculate the biomass of the classroom in grams per meter squared. So the amount of mass of, say, humans within that square classroom area. And grams per meter squared is the standard units of biomass, so it gives them that context. And then they think about how biomass would change if the mass of the organisms present changed. So if you added 50 more people, or if all of a sudden the same number of people went to the gymnasium and we measured the biomass there. So playing around with what biomass kind of means and what it looks like when we change the mass or change the area. Part 2 really gets them to think about vegetation itself in context of global biomes. So students rank global biomes from greatest to least biomass and compare their guess to available data. This is a good exercise, but might need some pre-teaching if students have never done any work with biomes before. There is a movie on the Globe Carbon Cycle page for the Globe website. So we're going to do something with this in just a moment. And then students after that typically try to estimate how much carbon storage is stored in their own schoolyard based on the biome classification that they came up with and what they observed in their field site. So we're going to go back to the Jamboard that you opened up earlier. And we're going to click at the top of the page on frame 2. The map will come up. I see some people have already started. So you're going to use the drawing tool to mark the location that you think has the highest plant biomass. You can see the key at the bottom tells you the different biome types. So if you're just coming in, you can click the pen over here on the edge. And then that allows you to mark where you think some of the highest plant biomass is in the world. We've got some guesses for the tropical and subtropical moist broadleaf. We've got a guess for the temperate conifer forests. The temperate broadleaf and mixed forests. If you've done the highest plant biomass, you can click to the third frame and work on the lowest plant biomass in the world. Where do you think that is? Which biomes have the lowest plant biomass in the world? Lots of activity here. I see people highlighting some of the world's deserts, some of the world's Arctic and Antarctic regions, some tropical savannas. So let's head back. You can kind of look at people's responses there, and we're going to look at another data source. The names of the biomes are slightly different because it comes from a different group of scientists. But if you take a look all the way down here, the highest biomass in the world is in tropical rainforests. So a lot of those areas that people did highlight on your maps. And that has 40,000 grams per meter squared of biomass. And then those of you who guessed rock, ice, and sand, you're right on the money. Only 20 grams per meter squared of biomass. Interestingly enough, compared to that desert area, or even the low tundra, which both have 600 grams per meter squared. So biomass across the world's biomes is quite drastically different. And so when you're studying carbon in your area, that'll be something you want to keep in mind, because the exact species that are there, as well as any human interference, could significantly impact your carbon storage and biomass number, your biomass and carbon storage numbers. So a thought exercise for you here. So consider, can you estimate how much carbon is stored in the sample site you described earlier? So the one that you described in that Padlet that you have already collected some data on, or you've been thinking about training or teaching at, how much biomass do you think it actually has? You'll want to consider what biome you're in here. You want to consider whether the site is a standard or non-standard site. So does it actually represent the biome that you live in or not? And then what are your actual observations of the site, and how do they differ from that natural biome? So College Woods lies in the temperate deciduous forest, but it also has a lot of evergreens. And someone did point out in that initial KWL that maybe softwoods and hardwoods have different characteristics that would change their carbon storage. And so I'm going to guess for College Woods, maybe between the two, maybe 30,000 grams per meter squared of biomass, and kind of store that in your mind, and we'll come back to that number a little bit later. So where do we go from here? We know biomass and carbon storage. We know DBH, but how do we get from DBH to biomass? Scientists use something called allometry. So how can you possibly measure dry mass of living plants in a given area without chopping them down, drying them, and weighing them? Because clearly you wouldn't want to do that if you're trying to keep the carbon in the vegetation. Over a long period of time, scientists, foresters, forest ecologists, researchers around the world have studied the relationships between parts of trees and their whole. And allometry generally is the relationship between the individual parts of a living organism and the whole of the organism. So for trees, there actually is a consistent relationship between diameter at breast height and the total biomass of the tree. And the exact relationship varies by tree species group. So it does vary slightly between each individual species, but there's a lot of commonalities between tree species groups. And we'll talk more about that in a second. And these are primarily due to wood density. So both cell layout patterns of the species as well as growth rates impact wood density, but wood density is the driver of this relationship. So in our activity, allometry, not a llama tree, there's also two parts. Again, the first part is an opportunity for students to explore the allometry of their own body, put it in that concrete world, and they actually get to create those mathematical relationships or allometric relationships for their foot length to their height and their arm span to their height. We're going to kind of gloss over that for now and head straight to the vegetation piece. And in part two, students use this concept of allometry to understand the relationship between DBH of trees and how that can be used to estimate biomass. So students are going to look at allometric relationships of tree species groups. So you can see that here in the graph on the right. This is called the predicted biomass graph, and it's a result of a meta-analysis of 100 different studies that took place in the United States. So 100 different studies that developed allometric relationships for trees in the United States was used to make this single graph. The author of the study, Jennifer Jenkins, and her team note in the beginning of their paper that this compilation of equations was needed to better understand biomass and carbon storage at the national scale. And this is important for our international participants, that other countries have completed similar national scale assessments. So if you're not from the United States, you may have relationships like these that have already been created by scientists in your home country. And likewise, if you live in a relatively temperate environment or have a lot of species, genuses in common with American trees, you can still use this graph, and we'll talk about that in a little bit. So just take a look at this graph. On the x-axis, we have DBH in centimeters from 0 to 50. And on the y-axis, we have predicted biomass in kilograms from 0 to 1600. And then these lines that you see on the graph, each line represents the relationship between DBH and predicted biomass of a tree species group. So if you look way up the steepest curve, group 1 is maple, oak, hickory, and beech. And then the shallowest curve, group 10, are woodland species. So just to make sure your brains are working today, if you went out and you measured a tree that was 30 centimeters in diameter, and it was in that maple, oak, hickory, beech group, group 1, what would its predicted biomass be? That's a question for you right now, and you can type your answer right into the chat. If you measured a tree that was in group 1 and its DBH was 30 centimeters, what would its predicted biomass be? People are trying to figure out how the graph works. See a couple of responses. So I can look at my 30 and kind of draw a line up to group 1. And then I can go across to the y-axis and see that, yes, in fact, a number of you have noted that it's 600 kilograms of predicted biomass for a group 1 species if it was measured at 30 centimeters. So a key point here is the steepness of the graph tells us something about the density of the tree. So if you're looking across here for group 1, we see that just a small increase in DBH has a larger increase in total biomass for group 1 than does, say, the woodland group or the cedar larch group, and so on. So density is the driver of this relationship. If your students haven't studied density before, we have a little extension to the allometry activity where you can take a look at that using wood blocks that you can just get at your local lumberyard or hardware store. So now we're going to take this and we're going to head into the data analysis piece. You can actually see this in action. Keep those questions coming. I notice Haley and Jen are working hard to answer all your questions. So here we are. We have gone out to our field site. We've measured tree circumference, and we know that it relates to DBH. We've participated in biomass units to understand this equation here. We've participated in the allometry activity to understand allometric equations. We know how big our sample site is, and now we're ready to do some data analysis. So most of you are familiar with entering data into the GLOBE website, and you can do that with carbon cycle data. But what happens when you put carbon data into the GLOBE website is it becomes kind of black box data. All of the calculations that we just talked about are hidden behind the scenes. And so we wanted to show you today kind of what's happening so that you have a better background when you go to train teachers or when you go to teach students of what's happening there so you can answer their questions. So here's our College Woods site again. Here we are, very, very many trees, very little undergrowth. And we will head out to our tree biomass analysis. Okay. So when you first open the analysis template on the GLOBE website, you'll see some things that you need to gather. You'll need to have the plot size and all of that sorted out. And then right here under data exploration are some details for how to explore this spreadsheet. So that's what I'm going to do with you right now. We're going to walk through it together so you have an idea of how to use this if you'd like. So our second tab along the bottom, you can find my cursor on your screen down at the bottom. And there's nothing for you to do except for follow along here. So second tab is plot size, and I've entered my 30 by 30 meter plot. The third tab is field data entry. So this is where you would enter the data you collected in the field. So here's the species group. You would have had to figure out already which species group each of your trees was in. And you would have given them a number and their measurement. So we're going to kind of fast forward a little bit to species group allometry. or actually, let's go all the way over to species groups. We're running a little short on time, so we're going to skip around a bit. So tab 8, species groups, here's that list of all the different species groups and the species within them. So if I use a find function, Command F in Mac, and I want to find out which species group my tree is in, I could type in paper birch, and I find that it comes up in the soft maple birch category, and it's a betula. So when I'm entering into that data entry section, I would put in soft maple birch as the species group. Now, I pointed out before, if you're an international participant, you might not be sure because you might not have exactly the same species. But we did ask you to collect some of that data. So I'm just going to give you an example here. Maybe. There we are. So let's just take a look here. From Argentina, we have an Acer rubra. So I can go back to my original thing, enter Acer. And I find that some of the maples are in the maple oak category and some of them aren't. So I might have to enter the whole name. And it doesn't find it, but we know that some of the maples are in the maple oak and some of them are in the soft maple birch. So that would be the point where you might want to seek out a local forester and talk to them about whether or not your maple tree is a hard maple or a soft maple, or find out more about its density. Is it very dense or is it not very dense? And likewise, that would be true. For example, I saw a number of trees from Thailand, which I know do not exist in the United States. So you'd have to reach out to those local foresters or forest ecologists in your area. So once you know a bit about the species groups, you can check out the species group allometry. Tab 6. And here's where the actual equations are. So right here, you can see the form of allometric equations that make up that exponential graph of biomass equals the exponent of coefficient B0 plus B1 times the natural log of the DBH. So all that's saying is that it's going to use these two coefficients in the equation. And then it's going to use your DBH. So we actually have an equation example if you wanted to see exactly what's happening in the spreadsheet, but the main pages are too confusing. So you can see this is how it's written in the spreadsheet, but you can also plug it into a calculator to back check it, which is a really interesting thing to do with especially high school students, because a lot of them are learning about base 10 logs and natural logs. So it's a great mathematical application that students can use. And here it is, they plugged it into a calculator exactly in this form and got 345 kilograms of biomass. And if you use the spreadsheet using the equation just plugged in here, B0 plus B1 times the natural log of B5, which was the DBH, you get the same thing. So it's just a way to experience what's happening with the data. So let's go back and look at our actual data. We'll come to the table summary in just a moment. So this is all your data in here. This is your original field data entry stuff that's just imported from the previous page, tab 3. Here is your calculated diameter. So that's your circumference divided by pi. And then here is that same equation, B0 plus B1 times the natural log of the DBH to get total above ground biomass for that singular tree. And here it is in grams, and then converted to carbon. So that's what's happening in behind the scenes on the Globe website when you enter just this one number here. It gets your individual tree carbon as the output. And then if you add up all your trees on your plot, you can get your total grams of biomass per plot and your total grams of carbon per plot. So if you were asking a question such as, how does the carbon on my plot compare to emissions in my schoolyard? And there were some students that did that. We might have time to show you that project quickly at the end. But then you can also use your sample site area or your plot size. You see that up here, plot size. It's dividing by that number to get grams per meter squared of biomass and grams of carbon per meter squared. So this is your total carbon storage by meter squared for your plot. And if you'll recall, this biomass number, 34,935, is actually pretty similar to what I estimated at the start when we were doing the biomass units activity. I estimated that College Woods would have about 30,000 grams per meter squared based on its biome. So that's a lot of information and we're almost out of time. So keep those questions coming in the Mentimeter so that you can get your questions answered in the protocol follow-up on Thursday. And we'll just take a few moments here now at the end to go back to our KWL. So lots of information in the chat about where to put your Mentimeter questions and when the protocol question and answer session is. And now back to your Jamboard. Back to frame one. And what did we learn? Take a moment to type in a few sticky notes. What did we learn about our question? How do differences in tree species and sample site type affect carbon storage near where I live? And if you see something in the center column that you just want to move and drag, feel free to do that also. Good, I see some things coming through that people have learned. The differences between standard and non-standard sites. That density has a big impact on biomass. Allometry is what allows us to make comparisons of carbon in different trees. The impact of species of the tree has on the amount of storage of carbon. Circumference and height of the trees. Conifers store less carbon than hardwoods. Density is key. So as you can see, you could probably ask lots of other questions using the same set of materials and still be able to answer them quite effectively. So you have access to this KWL for a little while here. We will refresh it for tomorrow's workshop, but feel free when people are done to take a screenshot of it on your browser. I just wanted to take a moment to let you know about a student project. Oops, sorry. A student project example on the Globe website. This student group was from Massachusetts. They're from Maynard High School, and they presented their research at the Globe SRS in 2019 in Boston. And their research question was, how does the uptake of carbon by schoolyard vegetation compare to the emissions of carbon by the school? So they used a whole different set of calculations to figure out emissions of carbon by the school, but then they used our materials to figure out how much vegetation, or how much carbon was stored in the vegetation in their schoolyard by doing a single plot and then scaling up to that school site. So I hope you all got a lot out of the workshop. I know it was so fast, which is why this will all be available in a recording for you to take a look at later. And we really hope you enjoy training in the e-training modules. Don't want to take up any more time today because I know you have other things to do.
Thanks so much, Sarah. That was wonderful. We have a break now until our next session, which will be a networking session. We have about 5 hours until that starts, so please feel free to check out our other meeting resources. This session will have the recording available on it on the meeting platform for you to access later. So thank you again so much, Sarah. That was wonderful, and have a great next few hours, everyone. Take care.
Thank you all for participating, and it was so nice to see some familiar faces and have a great full session.